All right, thank you guys for being here today, and I appreciate this. This is going to be a very interesting panel, and the topic of the panel is the future of vaccine science. So I want to, right here up front, just kind of talk about one thing. Number one, I want to make 3 points. Point number one, vaccines work. Point number two, vaccines save lives. And point number three, vaccines are the biggest public health initiative we've had in the last 75 plus years. Okay. Now that we have said that, I want to introduce our panel. What I'll do is a typical discussion. At the end, we'll have 15 plus minutes of questions and answers. You can certainly ask whatever you want, and they will answer the questions you have. But let me tell you who we have here. Vinod Balachandran. He is the founding director of the Oleon Center for Cancer Vaccines at Memorial Sloan Kettering Center. Thank you very much. Frances Lund. She's the director of Immunology Institute and the endowed chair at the University of Alabama at Birmingham. And Buddy Creech and I have a history back during the pandemic. I used him as one of my key resources on NBC. He's the director of the Vanderbilt Vaccine Research Program at the Vanderbilt University School of Medicine. Thank you all for being here today.
John.
It's going to be a great conversation. And let me start off with the big question that we call the 30,000-foot view. What is the future of vaccine science?
Why did you all look at me? That's great. All right, this is going to be this is a dumpster fire already. The future of vaccine science is really exciting because we have tools now that we did not have 15, 20 years ago to understand, decipher, and unlock the human immune system. We know more. We have better tools around DNA sequencing, around multi-omics technology. We have the ability with supercomputing to be able to now analyze that in speeds that we simply didn't have. And so right now, we're on the cusp of what I think would be a golden age of vaccinology, where we've tackled some of the low-hanging fruit, measles, mumps, rubella. Now we've got some of those big, difficult ones that have evaded us for years. Now we can get to those.
Yeah, just to add to that, I think specifically for cancer vaccines, cancer vaccines, the idea has been perhaps one of the most sought after challenges in modern, cancer medicine. Can we make vaccines against cancer? But we just have not really been able to have all the necessary components to be able to understand how would you do that. But to add to what Buddy is saying, we actually are in a moment in time where we actually have all of the knowledge and the technological advances to actually make effective cancer vaccines, turn it into a reality. So I agree with him that I think it's really exciting time for vaccinology.
Frances?
So I would say that, one of the big challenges that we've faced with infectious disease vaccines is how to make vaccines durable, how to make them last a lifetime, and how to broaden their ability to cover variants, for example, for viruses. And we're finally at a point now that we really understand enough about the immune system to understand what memory means and how to engage that through vaccinology to induce those cells that will last a lifetime that can protect you.
And just to give an idea of kind of the steps and how fast the steps have accelerated over the last few years, let's go back in time. Jenner, original vaccine. When was that? It was...
18th century is when we started doing all that. Yeah.
Right, and then
17th.
Vaccines started accelerating around the 1950s or so. That's when we really started accelerating vaccines, and they were stable for decades. Every now and then a new vaccine would come along. Has that process accelerated? Are we going to start seeing vaccines faster and faster, improvements in vaccines? Or does it depend?
So, John, some of that is going to be market forces that drive it. So there's a variable right now that we cannot control. The science for many of these is there. The public will or the private equity will to get that done will be a rate limiting step. And so I think that'll be an important piece of this. But I think you can look at what we've done around influenza, around COVID, around RSV vaccines. Maybe we don't think a lot about RSV unless we have a little baby at home, or we have our first grandchild now. So I'm thinking about RSV all the time during the winter because I know it's one of the most common reasons kids are admitted to the hospital. Well, because we understand the science of RSV now, this virus, we know that we can protect elderly adults. We know we can protect those who are younger, who have underlying health problems. We know that we can protect pregnant women, and we know that we can protect newborns. So all of a sudden, this virus that was our bread and butter in the hospital setting as a pediatrician, our residents don't know how to treat it as effectively because we don't see it as much. That's just one example of a vaccine that we didn't think was possible 6-7 years ago that we're now having the benefits from.
Francis, are you going to say anything? Or? No, you're
Nope,
good?
I was not.
Well, I think for specifically for cancer vaccines, it's really quite an exciting time because the immune system is essentially the most powerful cancer killing machine in the body, but we have not really understood how could we tap into this awesome force to be able to kill and eradicate cancers with minimal or less side effects than the current treatments that we have for cancer, which include chemotherapies and other things which have lots of side effects and really don't work very well. But we have the field has really struggled to understand how to do this, but now, due to advances really borrowing from what has been happening in the infectious disease field with vaccines and other advances, we are now in a position where we can actually really act upon this. So I think it's really exciting for cancer.
I was just going to add one other thing that I think we are on the cusp of being able to do, and that is for a lot of the infectious disease vaccines, they don't necessarily prevent you from being infected. What they prevent is morbidity and mortality, right? And that's critical, but the next step is to prevent transmission, right? That you have a vaccine that doesn't necessarily cause what we call sterilizing immunity. You never get infected. You get infected a little bit, you clear several days earlier, and so you're less sick and you have less days that you transmit. But if we could lower that distance and that time, then the likelihood that you will transmit to your baby in your house or your elderly parent goes way down, right? So that's a huge thing that I think we're really just at the edge of being able to do.
And diving kind of a bit more specifics, I'm a self-admitted needlephobe. I don't like shots. I get them all, especially I was in the military, so you get tons of shots, and most people don't. At the same time, I never really look forward to the flu vaccine. I don't know if anybody does. Are we making any headways on the universal flu vaccine, one that will cover flu for years? We don't have to get it all the time.
Let me answer that one first.
Well, so we're part of a national collaborative around universal flu vaccine development. And one of the reasons it's really, really difficult is first impressions matter. First impressions matter. That's true of our immune system too. So the very first time that you see a pathogen, it sets your immune system on a course that's very difficult to move away from. And the first person who described this around influenza was a guy named Tommy Francis in the 50s and 60s, whose dad was a Presbyterian minister. So he, of course, used that terminology and called it original sin or original antigenic sin, like a good Presbyterian would. And so we now call that antigenic imprinting, almost like a bird imprinting on their young. First impressions matter. So now that we understand that, we've now got some tools that we can try to overcome that. So how are we going to do that with influenza? In Europe for many years, and in the United States more recently, we've been using more potent adjuvants that are oil in water droplets. And these really tick off important cells in your deltoid muscle when you get the vaccine. And when we do that, we start to see a reworking of that original imprint that was laid down when you were a kid so that we can show you something and you care about it. That's going to get us one step closer to getting a universal vaccine that may not be perfect, but keeps you out of the hospital, keeps you from dying. I think that's going to be the goal.
And that is essentially the bottom line. Transmission is important, trying to cut down transmission. And I talked about this many times during the pandemic. The bottom line is keeping you from dying, keeping you out of the hospital, keeping you as healthy as possible. And vaccines do a fantastic job of doing that. Vinod, let me ask you a question, because during the pandemic, it was around early 2021, I was talking to one of the developers of the mRNA technology. And she was very excited about the potential, particularly the potential for cancer treatment. And the way she described it to me is, we now have a tool that we can attack the tumor. The big hurdle is going to be finding the antigen that we can put in this tool. Is that where we are still?
Yeah, so actually, to make an effective cancer vaccine, you essentially need 4 things. You need to find the right antigen. So this is the protein in the cancer that the immune system allows the immune system to recognize a cancer as foreign. I'll highlight that this is a slightly more bigger challenge because Unlike the immune system recognizing a virus or a bacteria where it is intrinsically not part of our body, the immune system is hardwired to recognize those things as foreign. Cancer derives from our own tissues, so the immune system is in fact hardwired to not recognize our own tissues as foreign. So to teach the immune system to recognize cancers as foreign, we have to find precisely the proteins that are found only in the cancer tissues but are not found in the normal tissues. So these particular antigens. What we have now learned over many years now, research from many different laboratories across the world, is that cancers develop because of breaks in the DNA or mutations. But as a cancer cell sort of divides and grows really rapidly, it accumulates more of these mutations or genetic errors, which allow the immune system to recognize cancers as foreign. These genetic errors are individual to every single person's cancer. So each person's immune system recognizes their cancer in a unique way compared to another individual. So what this means is that many of these vaccines have to be custom made.
So what works on you might not work on me.
That's right. So these vaccines have to be custom made based upon individual analysis of each person's cancer. So for this, you need a platform that can do this really quickly. So actually, our group ran a clinical trial doing this individualized cancer vaccines made with RNA technology because we thought RNA was always going to be really quickly. We were testing this on pancreas cancer, one of the most deadly cancers in oncology, and we had started this before the pandemic because we really thought the promise of this platform was really, really quite transformative. So yes, I think we now have, particularly with this current age of genetic medicines, of RNA, DNA, and also improvements in sequencing, et cetera, we can actually make bespoke vaccines for each cancer patient in real time. And this has been done. Our group did it. Many other groups have done it. So I think it is reality now. Yeah.
And a vaccine, and we actually talked about this when we had a call a couple weeks ago, a vaccine is only as good as being administered. So if it doesn't get into the person, that vaccine could be the best vaccine in the world, but it's not working. Are we working on administration techniques other than, again, I'm a needle phobic, other ways of administering it?
Definitely. So I think one of the big interests, at least in the context of respiratory viruses, is that you don't get infected in your arm, you get infected in your nose and in your lung, right? And so how do you develop an immune response that is in those sites where the virus is, and how do you maintain them there? And the shot in the arm actually doesn't do a great job of that. And the reason that it doesn't is that the immune system has been designed to have lymph nodes in all different parts of your body, and so the shot in the arm, the antigens that are in that vaccine go to the lymph node that drains that part of your arm, and that doesn't go to your lung. And so by coming up with RNA-based vaccines or the self-amplifying RNA-based vaccines or even some of the adenovirus vectored vaccines that can be delivered intranasally, that allows you to establish the immune response where you want it to be. And all of these things have come up in the last 10 years, maybe?
Yeah, I think that's right. And it's not that an injection in the arm does not work, but it doesn't have that durability that Fran was talking about earlier, and it doesn't necessarily have the ability to protect against those transmission events or local replication. So that's really a big effort from most vaccinologists right now, is if it's an enteric or intestinal pathogen, well, let's give the vaccine through the gut. And we already do that for things like typhoid or for rotavirus to prevent infant diarrhea. If it's something in the nose, like a respiratory virus or whooping cough, well, let's give it through the nose. We already have that for influenza. We've got development of that. We're starting a trial in a few months on a COVID vaccine that will be in the nose. And there are other efforts around pertussis, again, to be able to show your immune system this pathogen in a way that so closely mimics what it would look like in a wild type or more natural environment, and that lay down an imprint that you can then take advantage of later. So what I think the future may be is that for those who may not have significant risk factors for severe disease, we provide local immunity through vaccines that may not give you the highest level of antibodies that circulate around your body, but they give you great antibody locally. And then if you enter into a period where you're at higher risk, maybe we supplement that with an injected vaccine that really bumps those antibodies up very high.
What's the struggle there? Because we've been giving vaccines in the arm since the 1950s, basically. My favorite vaccine was when I was a kid, the polio vaccine, because you got it on a sugar cube. And I kept wanting to get more and more of those. Thankfully, my parents wouldn't let me. So what's the hurdle there to getting the intranasal vaccines, the gut vaccines like you talk about?
Well, some of it is that our bodies inactivate it before it can get to the business end of our immune system. So if you put a vaccine into the gut and your stomach acid deactivates it or inactivates it or kills it before it can actually reach those intestinal cells that are immunologically active, then you've lost. The second would be in the nose. If all you generate is local immunity without any sort of widespread, what we call systemic immunity in your bloodstream, then it may not be as effective in the long term. But I think that's where that 1-2 punch comes in, where we might be able to have our cake and eat it too. Give a intranasal or enteric vaccine and follow it during periods of high risk or in times where we need to have a boost with something that is either another dose in the nose or an injected vaccine. I think the biggest hurdle is presenting the antigen in a way to those cells that makes sense. We know how to do it in the arm. It's a little harder to do it locally.
So I think these improvements or technological innovations in platform design is actually extremely critical and will have cross-sector implications for both pathogen vaccines and cancer vaccines. So for example, the current generation of cancer vaccines that we were using with RNA vaccines are a little bit different from an RNA vaccine that you would give for COVID. So COVID vaccines, you get a shot in the arm. The current generation of cancer vaccines, it's actually an infusion through the vein. The reason is because for cancer vaccines, the thinking is you have to make a much stronger immune response to kill cancer cells as opposed to maybe you would need for SARS-CoV-2 or other pathogens. But this is V1, version 1 of cancer vaccines, and these iterations that you can have in platform design for intranasal or other routes of administration would then allow us to then repurpose or learn from that and apply them for cancer vaccines. So you could even envision a future where you'd say you go to, let's say, CVS and you get your cancer vaccine. You don't have to go to see your oncologist and actually sit in a chair for 4 hours getting an infusion, which is what currently cancer patients have to face with. So I think this is why we need continued progress and innovation in this sector. I think the opportunities are really, really quite transformative.
And I think, one of the reasons that for example, the intranasal vaccines sort of were backburnered for a long time is that you can't just put something in your nose and assume that your immune system's going to pay attention to it, right? You're inhaling stuff all the time, and 99.999% of it, the immune system ignores. So you have to put something in that will actually cue the immune system, but at the same time, not make it respond so robustly that you end up with the equivalent of an infection, it's not an infection,
right,
from your vaccine, right? You don't want a huge amount of inflammation. You want enough that your immune system pays attention to it and does something about it. And it took us a long time to get to that point there where we could have the appropriate kinds of vectors or adjuvants that would engage the immune system in a way that was not damaging, but still productive.
Well, I was going to say that a great case principle in this is the rotavirus vaccine that we developed in the late 90s, used it in kids, but it caused such local inflammation that very rarely kids developed a condition called intussusception, where the intestines telescoped over each other and it caused what would be a lack of blood flow to that place in the belly. That vaccine was immediately taken off the market because of that. Our vaccine safety systems worked, and it took years to figure out exactly what Fran was saying. Let's dial it back just enough that the safety of it is maintained, but let's make sure that that the immunogenicity of it is maintained, but that we recover the safety that we need. That's the same for influenza vaccine. We don't give intranasal vaccine to kids under 2 because it can sometimes make them wheeze, like an asthma attack. That's because it works differently in that age group, and we just have to dial that in, and that takes time.
From a perspective of experts here, looking back at the pandemic, 2020 timeframe, how impressive, or was it impressive, that we came up with a COVID vaccine that quickly?
It was
yeah,
insane.
it's insane. But yeah, you take it, and I'll add on.
Yeah, I mean, to think that we went from a virus first being identified in
January 2020, right?
to having a vaccine that we could administer to people less than 1 year later.
December, right.
That's unbelievable. I mean, and that was because, and so I'm going to make that pitch, the basic research that is funded through the federal government, through the NIH, allowed for the development of the RNA technologies that were used in the generation of those vaccines. The sequencing of those viruses, of the virus isolates that came very early on, was critical to designing what needed to be the antigen that went into the vaccine. All of that was done through basic science and really fundamental research that we've funded for the last 50 years. And we came together.
If you take nothing else, that is your takeaway right there. If nothing else leaves here, please let that be what comes. We did not make a COVID vaccine in 9 months. It took us like 9 months plus 20 years. And what happened in those 20 years? 3 things. Mark Dennison at Vanderbilt, Ralph Barrick at UNC figured out by studying mouse hepatitis virus. What the crap is that? It's for all those mice living hard out on the streets. They studied the basic virology. How does a virus get into a cell? So when the OG SARS came in 2001, they had reagents to use in the lab that nobody else did. And so when this next generation coronavirus comes along, mouse hepatitis virus, what kind of virus is it? It's a coronavirus. So they had the tools. That was stage 1. Stage 2 was Barney Graham and Jason McClellan at the Vaccine Research Center. They figured out that when a virus is docking with a cell, it starts out in this shape, and then it moves to this shape once it docks. And for years, we had been working on this shape to make vaccines. It's too late. It's already stuck to the cell. So they figured out a way to stabilize the important proteins on the surface so it stayed in this pre-fusion conformation. That was step 2. And then step 3 was Drew Weissman and his team at UPenn figuring out just how to make mRNA vaccines deliver what we wanted them to without being too reactive. Those 3 things took so much investment. And then once the US government through Operation Warp Speed said go, we went. And we had an infrastructure that could do it.
And did that, what we learned from that, and the technologies that came out of that, accelerate cancer vaccines?
Yeah, the story of cancer vaccines and RNA is quite interesting also because if you may know, actually the original company, BioNTech, who made RNA vaccines for SARS-CoV-2, actually started off as a cancer vaccine company. The idea was to make RNA vaccines to treat cancer, and because of the platform having really a lot of, the rapidity of the platform, you can essentially make a cancer vaccine really quickly with RNA, and this was important because for cancer, these vaccines needed to be individualized. So, for instance, we had been working on developing cancer vaccines for pancreas cancer, and we had started with BioNTech actually a clinical trial treating pancreas cancer patients with RNA vaccines before the pandemic. So in fact, some of the earliest patients who were getting an RNA vaccine for anything on the East Coast were patients getting RNA vaccines for cancer, actually not for SARS-CoV-2 at all. And this was in December of 2019. And then the pandemic happened, so it was actually, so then they had to actually pivot, and because we all knew the platform was so good, so they pivoted to be able to utilize the platform for making infectious disease vaccines. So it was sort of an acceleration of the effort. So
and then since then,
well, we've learned so many things from that. So because we had always envisioned that the platform would be very safe, but in a small clinical trial or a smaller clinical trial on cancer patients. Although we found it was very safe, we had a lot more information on the safety of the platform when it got applied globally at scales that we could not imagine for SARS-CoV-2. So it was like, oh, okay, it is really safe. And oh, yeah, it really works really well. So our thinking was correct that the platform is really good, can really make a very powerful immune response suitable for treatment for cancer. So I think it was sort of a collision of 2 areas of research in a very interesting time then, yeah.
Oh, great. And one of the things is, this new technology, the RNA vaccines, doesn't mean we give up on the old technology.
No.
There's still utility, and in some cases, it might even be superior. Is that correct? Or it depends?
I think it depends on what you're trying to accomplish. So I think every vaccine should be designed to fit the disease you're trying to eradicate or cure. And I think RNA-based vaccines, the beauty of those is that you can make them very, very quickly. And so for cancer, that's going to be critical. For pandemics, it's going to be critical. But, you know, I don't know that we need to change our flu vaccine to an RNA-based vaccine. We need to think about how we deliver our flu vaccine, and maybe it will turn out that RNA is the best way to deliver it. But our trick with flu vaccines is how do we deliver it in the right way to get the response that we want in the tissue that we want. And that may be RNA, but it could just as easily be some of these vectored vaccines that look like a virus, but are not a virus, that the immune system is going to respond to.
And one of the things the FDA has done for years, which I really respect, is that when they license or authorize a vaccine, it's not a blanket. It's that individual component, that individual construct is what's approved. And the reason I bring that up is that there are some times when we start talking about mRNA vaccines and we treat them like they're all the same. And they're really not. An mRNA vaccine that targets COVID is going to have a different safety profile, potentially, than an mRNA vaccine for flu. Might have different duration of immunity. Why? Because all the mRNA tech is doing is taking the protein manufacturing plant from Kalamazoo, Michigan, to your deltoid. I mean, unless you live in Kalamazoo, Michigan, then it's still in Kalamazoo, but that's a different thing. So all we're doing is putting it in your deltoid. And some of the side effect profiles that we saw from the mRNA COVID vaccine, like heart inflammation in young adult males, we think that's really something that we have to focus on with SARS coronavirus, not the mRNA platform in general, but we have to study that and we have to evaluate it. I think one of the most important things that you should hear us say is that the future of vaccine development can never be severed from vaccine safety initiatives, vaccine effectiveness issues, and the basic science that drives us.
And I would also like to say that, you know, one of the things with RNA-based vaccines, or at least the first generation of them, is, you know, they have to be kept cold. Well, that really changes it in terms of how do you deliver it to
And where and
places. And, you know, so some of the things that are coming on from that are now, they have RNA technology as a part of it, but a lot of it is designed to be something that can be at room temperature, that's stable for months, et cetera. And all of those things, I think, are going to be those second and third generations that come out of this and will make that vaccine a lot more effective and a lot more usable worldwide, globally.
And for cancer vaccines, are there any old technologies that might work, might help out? Because we're talking about RNA, but are there other technologies?
Well, I think among the most promising delivery platforms for cancer vaccines are the genetic platforms, the RNA and potentially DNA-based platforms, for a variety of reasons. One main one being the speed. You know, you can basically generate an individualized vaccine for a patient in weeks, and this can be administered safely and generate an immune response which could potentially impact outcome. And I just want to highlight that for cancer, you know, cancer remains second leading cause of death in all Americans. 1 in 3 Americans will face cancer throughout their lifetime. And in the next 25 years, this incidence is expected to increase by about 50%. Oh. And the current treatments that we use are all static treatments, so meaning chemotherapies. Once you stop the medicine, it stops working. But we've always envisioned, can we develop a living medicine for a one and done for patients with cancer? Limited treatment for lifetime protection and cure. Such potential only happens with the immune system. This is the only way to do it. And one of the most powerful ways that we have used the immune system to fight any disease is a vaccine. So we are now at the cusp of really being able to repurpose this for really potentially transformative impact for patients who really need it. 100% of patients who get a cancer vaccine have cancer. And their other choices are bad. So you know, I think it's a real responsibility for us to continue forward here.
That's amazing. But when we look at the COVID vaccine that came along, and we talked about how that was less than a year to get it to from one point to where it's being administered. Did a lot of that have to do with data sharing? As in, was that a time period where data was freely shared versus other time periods where it's harder to get data shared across platforms, across entities, across companies?
Yeah, I don't know how much of it was data sharing. Some of it was for any engineer, you're about to get so excited. Instead of working in series, we worked in parallel. See, there's a couple that you got really excited. What we usually do is we do a phase 1 and then we pause. And then we have some people meet about it and we talk about it and we try to get investment to go to phase, and then you do phase 2. And then you get the data and you wait for investments and you wait for data analysis and then you go to phase 3. For this, because speed was of the essence, the Biden administration deserves a lot of credit for rolling out vaccine. The Trump administration deserves a lot of credit for putting all of our noses in the same direction, saying, I don't care what you work on, you're working on COVID right now. And what that allowed us to do is we started the phase 1s while writing the phase 2, 3 protocols, doing it at risk because maybe the phase 1 wouldn't work. When it worked and we got a dose, we already had the protocol ready to go. And then we leveraged our rich infrastructure of clinical trial sites around the United States, like the vaccine and treatment evaluation units that there are 10 of in the country, and the HIV networks, and we all did this. And so enrolling 30,000 people sounds daunting unless you're doing it with literally all your friends at the same time. And that's what we were able to do. So we didn't rush the process of getting a vaccine from stem to stern. We just did it in one of the most brilliant ways possible by pointing all of our noses in the same direction. So there were data sharing elements, but not really between US government and Pfizer, because they were running their own separate program outside of that. It was really a matter of everyone had a call to arms moment, and we responded.
But I would say that there was a lot of data sharing around the virus itself, right?
Sure.
And trying to understand, what did the virus look like? What was the sequence of the virus? What were the variants, et cetera? And all of that informed the vaccine design.
That's very true.
And all of that data was absolutely shared across the world.
And now? Is that sharing still there?
It is academically. I can't speak to a worldwide phenomenon of what that looks like, but I have not lacked for data when needing it. But that's an N of 1, so you shouldn't do much with that.
Yeah, but I think, another thing that the government invested in wisely, I think, over the years has been these repositories. We have many, many repositories where our data goes so that it can be shared. And that is expensive to maintain those repositories and to curate them and to make that data available. And that's a fear, right? Is that going to stay? And I really hope it does, but will it?
Same concerns?
I mean, I think the lesson here, really, with the remarkable success of creation of SARS-CoV-2 vaccines in record time, is what can the academic, what can all the sectors achieve when we come together? So federal, philanthropy, academia, and industry, and they all came really spectacularly together to solve this problem. Now, how can we harness this to tackle other problems such as cancer, for example? How much could we achieve if we could actually get together and do this? I think we could achieve quite a lot. So I think that's really, what I have, sort of viewed in awe at how the community came together with this. And was like, oh, how can we get this together for now cancer, for other really, unprecedented challenges for the country, and I think this just shows that what we can do when we
replicate that.
Yeah, when we work together. Yeah.
Now, one of we understand, science is accelerating. Science is doing great leaps in looking at these vaccines and different platforms and different ways of administering. Is science outpacing public trust, or is science interfering with public? Because again, the vaccines are only as good as people taking the vaccines.
Yeah.
Any issues or concerns there?
Well, so as scientists, we're often terrible communicators. We're often terrible communicators.
Not physicians, we're the best.
We also tend to suck. I don't know if you can say suck in Aspen, but I just did. Sorry for that. We sometimes are just very difficult. We sometimes either downplay successes or we overplay other things. Sometimes we can be too paternalistic. Sometimes we don't take the time to actually sit and talk to folks, right? I need, as a pediatrician, to hear your concerns as a parent about what you're worried about with vaccinating your children. I need to sit and engage with you on that. If I don't do that, how are we going to have a trustworthy relationship? We're not. If I expect that of the guy trying to fix my brakes on my car, you should be able to trust me to do that as your physician. So one of it is just communication in general. The second is we've got to restore, I think, the trustworthiness of the systems that we have. And I think there's a lot of noise out there right now about how folks are conflicted, or how if you talk with someone from industry, you can't make an independent decision. And I think we just need to cut through that and say, no, what we really need is everyone talking to each other. Where we need to recuse, we can, but let's make sure that we're in relationship with each other. So those would be my two things: communicate better and build relationships.
But I think we have to have better outreach. So communication is a part of it, but where do I engage with the community? Where do I belong? Where should I be sitting so that I can answer questions or that I can ask questions? And I don't feel like we have that town hall. And I think that town hall is really important. And I think that that's a place where academia could do better by, instead of being insular, here we are in this institution, is here we are in an institution, we are in a community, we are in a state. How do we engage with the people that we are supposed to be serving? And I think that's one of the things that scientists are not trained to do that. We are, at least in my lab, we're working on that now. And every time we give lab meeting, we make the person who's giving lab meeting that day stand up and in 2 minutes explain to their mother, their grandmother, whoever, what they're doing and why it's important. And I think that's what we need to learn how to do better.
Perfect.
Yeah, one thing we've been stressing for cancer vaccines is really that these medicines are for patients, all of whom who have a diagnosis of cancer. Most of them have a diagnosis of a deadly cancer. So they are facing death. That's what they are, what they are hearing from their physicians, And our goal with these treatments is to hopefully get them another option, a better option than what we have currently, and hopefully get to a point where we can even talk about in future the C word, the cure word. So I think this is really what we've been trying to stress for cancer vaccines, that these we think will be important medicines which will give hopefully good alternatives for patients who have really currently not very effective treatments.
Fantastic. I do want to open it up to questions from the audience. So way in the back there, right there.
Hi, I'm wondering why cancer vaccines are really not so readily available in the U.S. and why so many patients are going to Germany where it has been available for the last decade.
Yeah. So right now, this is partially because most of the cancer vaccines are in clinical trials as of now. We do not, in fact, have broad FDA approvals of this current slate of next generation cancer vaccines in the United States. So the current evidence indicates that these vaccines are potentially effective and it looks very promising, but they have not progressed for FDA approval yet. Why are they available in Germany? I can't really exactly answer that question, but I can tell you that I get this question a lot from my own patients and many other cancer patients who ask the same thing. What I would say is that, our thinking behind cancer vaccines here is we want to make sure that before you get to FDA approval, we ensure all of the critical components that you would need for a medicine to be approved, namely safety and efficacy. They need to be safe, and we have to prove that they work. So until we can get there, we won't actually get to an approval. So.
Sir, right here in front.
So how do you think the composition of the current vaccine advisory committee is going to affect not only R&D but public trust?
This is the international symbol for not it. Yeah. I can start. One of my friends was chair of the ACIP, Kip Talbot, maybe I'll make two comments. One, the 17 individuals that were on there prior deserve a lot of respect for the work that they did. and they were incredibly talented, and they have a specific expertise. I don't know but one of the new 8 ACIP folks, and I don't know what that looks like. I can't yet ascertain on paper what the specific expertise will be and how that will drive the decision making. But I can't make that decision because I don't know them, and that would be unfair. What I know is it's causing a lot of upheaval and fear for those manufacturers who have products that are right there on the cusp. And so maybe I'd finish with one thing. It has been our strategy up till now that the ACIP makes recommendations that all of us as physicians follow and the payers have agreed to. It may be that the future is a different type of advisory board that is not part of CDC, that's made up of our professional organizations that has that same effect. There's a lot of folks thinking about it. So the answer is I don't know, but I know the 17 very well, and they were really good.
Yeah.
Anybody else want to take a shot?
They were really good. That's really sad.
Another question in the back there?
Thank you, and thank you for this
I
wonderful.
won't ignore this slide, I promise.
Sort of related to your question about the availability in Germany, as a parent thinking about whether I have to take my child to Canada in the fall to get her seasonal COVID and flu vaccines, I'm thinking a lot lately about international vaccine, programs. You talked a bit about how the cuts are going to impact basic research and presumably the pace of some of the development you guys were talking about. How integrated or not are our research programs in vaccines and those in other countries? Are approval systems in those in other countries? Like, can we hope that progress will maybe be made faster in other places in the next 4 years and we can capitalize on that as Americans?
Yeah, I can speak to it a little bit. I mean, we finished an mpox-based vaccine study to make sure that we knew the safety and immunogenicity profile in teenagers because of the ongoing outbreaks in the DRC. And the first group to act on that was the EMA. So we already know that Europe will rely on some of the science that we do in order to build out a package. I would assume that the reverse could be true, although there are our own Code of Federal Regulations and laws that would govern how much we can import from a data standpoint. So I think there will be some legal challenges with that. I think the academic community right now is currently global in scope. I think that gets challenging. You could predict it goes one of two ways. Either our ability to connect will be challenged and it won't be as good, or quite frankly, the number of US scientists that are leaving to Europe and to Australia and to Canada is probably going to increase at a rate that is disturbing to us if we're going to continue to lead biomedical research. Whether that will end up helping because we know each other or continuing to deepen the divide, I don't know. What I hope, though, is that at the end of the day, parental choice and the ability to vaccinate our children won't be taken away from us, right? You should never have to leave our country to get an FDA-licensed vaccine. And so what I hope is that when all the dust settles, maybe there will be different recommendations and strength of recommendations, not a mandate, but an encouragement. I hope that's where it lands.
And I would just say on the basic side, while there is beautiful work done in many other countries, the United States leads the world currently in basic science, and that's because we've made the investment and because we had the capacity to do that. If that goes away here, there isn't somebody immediately that's going to step up or can step up to replace that. And so I think that sort of building block section that we think about that is going to change health in 20 years, we could lose a generation of that if we go away and there isn't somebody else that can step into.
Yeah, for cancer vaccines specifically, I would say that the infrastructure here we have for cancer research in the United States really, is unparalleled and outpaces essentially all other, countries, I would say, in the world. So I agree that if we don't do it, I don't think others will. But I am very hopeful, actually, for cancer vaccines because just what is appearing on the horizon for what these new transformative technologies could mean for patients who have really currently not very good outcomes is so compelling. How could we not do this? We would have to do this. We're talking about potentially life or death for people, for so many people. So I think I'm very hopeful actually that we will, now is not the time to slow down. Now is the time to go full gas, that we really have to make investments in this space. We really have to really take the mantle, because I think this can lead to, huge improvements in survival, cure for people around the world, yeah.
And it may be that it actually goes the opposite, that the money goes and flows to the cancer vaccines for a while, and we learn in the infectious disease community, we learn from that. And when the time is right, maybe we can take what they develop and push it back into our space, right? And that's the beautiful part about the sort of interdisciplinary nature of the science.
Here, Bob.
Thank you very much for a wonderful discussion. I'm Bob Wales, a retired physician. What I've heard from my colleagues is there's real changes right now. So it's kind of a corollary to what you were just talking about. My question is, can you give me examples within your own systems on the impacts already on the NIH cuts in funding?
Yeah, I can start. The biggest impacts on trainees. Our ability to fund postdoctoral students, postdoctoral fellows, graduate students, MD, PhD programs, that has really taken an enormous cut. So that's number one, is that we're going to lose a generation of scientists because we can't figure out how to pay for them. And one option would be to, well, pull from endowments or pull from philanthropy. That's great. We already use that to keep the lights on and to pay for plumbing and all of that. So it's not an easy fix. That's number one where we felt it. Number two where we felt it is that right now we're hitting a common storm of NIH funding is at risk and Medicare, Medicaid funding is at risk. And so our systems are collapsing onto each other to say, we need to save money. Not $50 million, not $100 million, but like $500 million. That's hard to do. Vanderbilt just announced that we were letting go 500-600 people because of an estimated shortfall of several $100 million. That's hard to do and keep up our mission of teaching, caring for patients, and discovery. So we're going to see disruptions across all those sectors. We take care of patients by the discoveries we make. We have discoveries because we have trainees. Those trainees are motivated by the patients they see. It's all a circle, and that three-legged stool is wobbly right now.
And I would say that, I come from a public institution in a very poor state. And so the resources we have are limited, but the people that we serve have great need. Mm-hmm. And so what goes when the resources go are the people that need the help.
Yeah, I would just echo that, the fields are all interconnected. So any impact on related fields can slow down the entire enterprise. So I think particularly for cancer vaccines, I think since the time is so exciting right now, because we can really potentially do something that we have been trying to do for over 100 years, that now is really the time to really make large investments and really move light speed ahead.
All right. One last question. This ma'am right here.
Hi, thank you. I'm Mary Woolley from Research America. And contrary to what was said and also implied, you're all great communicators. Let me just say that.
Thank you for that.
And valuing communication, such as you've been displaying, is too often missing in academia. There isn't enough attention to it. You spoke a little bit, Fran, about how you're doing things in your own lab. But we need more of that in academia, number one. But I also wonder if you would agree with me that we also need more scientists, more clinicians to be communicating with their elected officials and urging others to do the same. Because we're not finished yet, including proposed budget cuts that haven't been enacted yet, even though many other cuts have happened. Some of those will be reversed, I believe, by the courts. But significant damage has already been done, but we don't need to keep it going. And that takes many more of us reaching out to the people who make the decision. So I'd love to know what you think about that and what you're doing.
Well, if Senator Blackburn from Tennessee has not put me on her do not email list, that's on her, because I've emailed her a ton and left a lot of voicemails. And I think the hard part is that we don't yet have a venue like this where we can just talk. We talk at each other with sound bites. We talk at each other with tweets or X's or whatever they are now. I'm too old. We talk at, not with. We've got to seek first to understand and then to be understood. There are a lot of core principles that we're missing right now, to Fran's point of we don't have a town hall. We speak in 140 characters or 35-second sound bites. This is complicated. We need minutes and hours and days with each other. If this group can figure out the venue to do that, we might be able to make some differences.
Yeah. And I think, so in the state of Alabama, we are thinking about how do we communicate with our legislators and how do we communicate with our neighbors. And I think that the tweets are not productive most of the time. And so we really need forums, like Buddy said, where we can talk about it, where you can say, well, I really don't think I should get my kid the measles vaccine. And we could say, well, why not? And somebody could explain why they think that's a bad idea. And we could say, well, have you thought about this, right? Not, well, that's dumb, you should do this, but more, well, have you thought about these other things? You don't get that opportunity. And that's what we really need.
Yeah, I think most academic institutions have realized this perhaps more so of late and are engaging through all channels available. Each institution has usually a robust government affairs group where they talk to people, and I think a lot of the individual investigators also have to really try to reach out, talk to their elected representatives, explain to them about why what they're doing is important, what is the implication for people, and trying to do this in a really clear way. And I think this is also why I think many of us attend these types of other forums, because we think, like you said, communication is extremely important, especially now. So we have to really explain why we think it's important so that others will, hopefully try to have a discussion about it.
And we're grateful for people like John who can coax that out of us, because we need that.
Yeah.
Thanks. And I personally want to thank the three of you and everybody else that's doing research for keeping the fires burning, keeping the candles lit, and keeping hope eternal. Thank you guys.